Hello students, a warm welcome to all of you in ITLS Academy. In today's lecture, we will discuss about the introduction of proteomics. Basically, in this video lecture, we will discuss about the what is proteomics, what is the techniques used in proteomics, and what is the application of proteomics. Okay, so we start today's lecture. First, we understand what is a proteomics. Basically, proteomics, we study the we study about the protein. We study about the protein. Okay, and this protein is a very essential for any living organism. Living organism. Okay, while genomics focus on a DNA and gene and protein. Okay, in in proteomics we study about the protein, but in genomics we study about the DNA or gene. Protein is concerned with the structure, functional and interaction of protein, the final product of the gene expression. Okay, in genomics we study about the DNA. In DNA, basically gene expression. Gene expression. And final result or outcome of gene expression is protein formation. And in proteomics, we study about this protein. This genomics and proteomics are interlinked with each other. Okay. In proteomics, we study about the structure, function, and interaction of protein with other molecule or protein-protein interactions. Okay. Since protein are responsible for virtual and every biological process, understanding their role in health, disease, and cellular function is a central to biological research and medicine. Protein involve This proteomics involves analyzing the complete set of protein expressed by the organisms at a given time. Okay. Now, the definition of proteomics involves the study, structure, expression, interaction, and cellular role of all the protein present in the particular organisms or proteome. Okay, what is a proteomics? Proteomics is a study about the what is a protein? What is a proteomics? Proteomics involves the study related to the structure, expression, interaction, and cellular role of the protein present within a particular organisms or proteome. Okay, now we understand the role of bioinformatics in proteomics. Proteomics in bioinformatics refers the application of tool computational tool, algorithm, and software to analyzing and interpret the vast amount of protein related data, and this protein related data is generated via the different techniques that are used in a proteomics. In bioinformatics, basically we analyzing the data that are generated by the proteomics technique. Okay, With the advent with the advent of high throughput technology like mass spectroscopy, protein microarray, the amount of protein data is grow exponentially, making the bioinformatics essential for managing, analyzing, and driving meaningful biological insights. In bioinformatics, basically, we manage, we analyze, and we interpret the data which are generated via the proteomics techniques, basically high throughput proteomic techniques such as mass spectroscopy and protein microarray. Okay, it also involves the application of bioinformatic tool and algorithm to extract the meaningful information from the proteomic experiment. Okay, this is the role of bioinformatics in a field of proteomics. Okay, now we discuss the key areas of proteomics in bioinformatics. First is data analysis. Processing and analyzing raw proteomic data such as mass spectroscopy data to identify the protein and estimate their abundance. Okay. Next, protein identification. Using the computational method to search the protein database to identify the protein based on their mass spectra. For the data analysis, we use bioinformatics tools and algorithms. For protein identification, we also use computational tools or computational method. Next, protein quantification. Estimating the relative abundance of protein in a sample using the computational method. For protein structure prediction, for predicting the protein's secondary and tertiary structure, bioinformatics plays a most important role. In bioinformatics, there are lots of tools and algorithms are available for identification of or to prediction of secondary and tertiary structure of protein. Next, protein and protein interactions. Okay, to understand the protein and protein interactions, presently we use a computational method. Okay, next, pathway analysis. To identify to identify the biological pathway involved in cellular process based on a proteomics data. Okay, next, system biology. Okay, for analyzing the data, for protein identification, for quantification of protein, structure prediction of protein to know the protein-protein interactions, pathway analysis, systems biology. In all these fields, protein analysis. So first, protein data analysis, protein identification, protein qualification, protein structure detection, both primary and secondary, to predict the Ramachandran plot, to determine the interaction between the protein and protein and with protein with other molecules also, pathway related to the proteins and system biology. In all of these field, in all of these areas, bioinformatics plays a most important role. In bioinformatics, there are a lots of algorithm and tools are available to perform these activities. Okay, we discuss the key aspect of proteomics in bioinformatics. First, protein identification. In a typical proteomics workflow, proteins are digested into the peptides and these peptides are analyzed using the mass spectroscopy. Then the result of mass spectroscopy must be matched via the protein databases. Okay, now Now we understand the role of bioinformatics for the protein identification. In bioinformatics, there are lots of tools are available to compare the experimental mass spectroscopy data with theoretical spectra from protein database to identify the protein. Okay. For comparison, the experimental MSG data with theoretical spectra of protein data to identify. For the comparing the experimental MSG data, we use mascot. For comparing the experimental MS data in bioinformatics, we use mascot, sequest, and extender. These are the tools which are used to compare the MS data with protein databases. Protein databases. Protein databases such as Uniprot or NCBI. Okay, next. De novo sequencing. Algorithms are used where no database is matched exist, then they try to interfere the protein sequence as identify protein sequence directly from the MS database. Okay. For identification of protein, for the identification of protein, some biological tools are also available, and some de novo sequencing technique is also available to understand the identification of protein. Protein quantification. Okay. Bioinformatics tools are used to quantify the amount of protein in a sample by analyzing the intensity of mass spectroscopy signal. For quantification of protein, we quantify the amount of protein in a given sample via mass spectroscopy signal. There are a lots of techniques also available to quantify the amount of protein. Such techniques are label-free quantification, isotope labeling. The data generated by the label-free quantification and isotope labeling. Okay, first we generate the data using the technique label-free quantification and isotope labeling. Okay, then this generated data is analyzed by the bioinformatics tool. Bioinformatics tools and software such as MaxQuant, Proteome Discover, and Skyline. Okay. First, we generate the data by the help of these techniques. Then we analyze this data by the help of bioinformatics tools and software. And these are the bioinformatics tools and software. Okay. Now we understand the post-translation modification analysis. Okay. Protein undergoes various post-translation modification. Okay, such as phosphorylation, glycosylation, ubiquitination, etc. And these post-translation modification plays a most important role to regulate the function of protein. Detecting these modifications from the data is very complex. Okay. In this scenario, bioinformatics tools play a most important role. For example, a specialized algorithm and tools like Phosphocyte Plus, PTM scan are used to identify and map the protein translation modification based on mass shift detected in the mass data. Okay, these are the tools which are used to identify the post translation modification, and this data is generated by the mass spectroscopy. The data generated by the mass spectroscopy is analyzed by this software. Then this software is very helpful to understand this post translation modification. Okay, next, post translation modification databases such as database post translation modification and Unimod help in annotating their identified modification. Then this database is very helpful to understand the modification. Okay, these are the tools which use to identify the modification, and these are the database that are used to identify the modification. Okay, now next we understand the protein structure. To determining the 3D structure of protein, it is very essential to understand their function. But experimental methods such as X-ray crystallography, NMR, etc. are laborious. Okay, but bioinformatics tool play a most important role for the predicting the for predicting the structure of protein. By the help of by the help of bioinformatic tools, we easily predict the secondary and tertiary structure of protein. Tools like AlphaFold, iTeaser, and Modeler. These are the tools which play a most important role to understand the protein structure. These are the tools which are used to predict the protein structure. These are the tools which are used to predict the protein structure. Rather than these tools, Chow-Fasman method, Gore method, this algorithm is also used to predict the secondary structure of protein. For constructing the 3D structure of protein, we also use the homology modeling. Homology modeling. Okay, next, database like PDB, that is a protein database, it store the experimental determined protein structure, which bioinformatics tool can be analyzed for structural similarity and functional annotation. Okay, PDB is the database for protein structure. Next, protein-protein interaction. Protein rarely work in isolation. They interact with other protein to form a complex and carry out the cellular process. Okay, most of the protein will interact with another protein or another molecules to perform their activity. Protein-protein databases such as String, BioGrid, and Intact store known interactions. Okay, these are the databases of protein-protein interactions. In this database, protein-protein databases are stored. Okay, tools like Cryptoscape allow for the visualization analysis of complex protein interactions network. Okay, these are the databases, and these are the Cryptoscape is a tool that allow for the visualization and analysis of complex protein interactions network. Okay, Computational methods like docking algorithm, coevolution analysis are used to predict the new interactions. Okay, this is the database. Okay, this is the tool, and this is the algorithm which is used to analyze the new protein-protein interactions. Okay, now we discuss the functional annotation of protein. When proteins are identified, they need to be annotated with functional information. Okay, such as their role in cellular pathway, enzymatic activity, or involvement in disease. Okay, for understanding the functional annotation of protein in bioinformatics, there are lots of tools are available, such as Blast2Go. Interproscan is used to predict the protein functional, and this is based on a sequence similarity and domain structure. These are the tools which are used to identify or annotate the protein function. Next, gene anatomy. Database. This is a type of database which provide a standardized vocabulary for describing the protein functional, biological process and cellular location. Now we understand the proteomics data integration. Proteomics data can be integrated with other omics data such as genomics, transcriptomics, and metabolism. Metabolism to gain a more holistic understanding of biological system. Okay, this proteomics data can be integrated with the other omics data, and these omics data may be genomics, transcriptomics, metabolomics, etc. Okay, now the role of bioinformatics in the proteomic data integration. Pathway like omicsnet, pathway commons facilitate the multi-omics data integration. These allow the researcher to understand how the changes in protein expression related to genetic mutation and metabolic shift. Pathway enrichment analysis tools like David, Reactome, and KHKE map the protein to know the biological pathway to identify over representative pathway or network in a data set. Okay, now we understand the data resources of proteomics. First is a UniProt, Pride, Peptide Atlas, and Human Protein Atlas. These are the database resources of proteomics. In UniProt is a comprehension database for protein sequence and functional information. This PRIDE, that is a proteomics identification database, stores the mass spectroscopy based proteomic data. In Peptide Atlas or Human Protein Atlas provide the data distributed and abundance of protein in different tissues and cell types. Okay. Next, for data visualization. In proteomics, tools are essential for the visualization of proteomic data, especially large-scale data set to identify trending pattern and outliers. Okay, bioinformatics tool. For the visualization tools like heat maps, Volcanoplot, protein interaction network are used to represent the proteomic data visually. Tools like CryptoCap or Python library are widely used to create a custom visualization and network graph. These tools are used to create the custom visualization as well as network map. Why we study proteomics? We study the proteomics to understand the protein function, to understand the disease biomarker, to develop the drug. These are the main role for study the proteomics. Now we understand the type of proteomics study. First is the expression proteomics. Second is the structural proteomics. The aim of expression proteomics expression proteomics to quantify the expression level of protein in different sample. Okay, in different sample to identify the protein that are up or down regulated in a particular conditions. Okay, this the aim of this expression proteomics is to quantify the expression level of protein in different sample. Okay, to identify the protein that are up or down regulated in a particular condition. Techniques used in expression proteomics: 2D gel electrophoresis, mass spectroscopy, protein level methods like ITR, isobaric tag for relative and absolute quantitation. Okay, next structural proteomics. Structural proteomics to focus to determine the 3D structure of protein and protein complex. Okay, for understanding the protein, to understand the protein structure, here we use the technique that is X-ray crystallography, nuclear magnetic resonance, cryptoelectron microscopy, etc. In expression proteomics, we use 2D gel electrophoresis, mass spectroscopy, and protein level method. And in structural proteomics, we use X-ray crystallography and NMR or cryptoelectron microscopy. These are the techniques which are used in proteomics. First is a mass spectroscopy. Mass spectroscopy is a core technology used in a proteomics. It will help to identify and quantify the protein by measuring the mass to charge ratio of peptide. Okay, and this peptide chain is derived from the protein. These are the basic steps of mass spectroscopy. Okay, next technique is a 2D gel electrophoresis. In 2D gel electrophoresis, basically we separate the proteins on the basis of their isoelectric point and molecular weight. Next, protein microarray. Protein microarray allow the study of thousands of proteins simultaneously. Next, Western protein. Okay, this Western protein is also used to detect and quantify the specific protein in a sample. These are the techniques which are used in proteomics. Now we discuss the challenges in proteomics bioinformatics. First is the data complexity. Proteome data is more complex than the genomic data because one gene is responsible this one gene is responsible for producing the multiple protein. Okay, thus this bioinformatics tools must account this complexity. Next, dynamic range. Proteomic data often contain a protein with vastly different expression level. Detect low abundance molecule against the backdrop of high abundance one of the challenging requiring the sophisticated algorithm to extract the meaningful data. Next, data standardization. With multiple experiment method like different mass spectroscopy setup are derived database standardizing proteomic data for constant interpretation across the study remain a challenge. Format like MZML, MZ Identity ML aim to standardize the proteomic data for easier sharing and analysis. Next, integration of data. Combining the proteomics data with genomics, transcriptomics, metabolome data create a system biology perspective require complex integration method and computational resources. These are the challenges in proteomics bioinformatics. Now we understand the application of proteomics. First application in the field of disease diagnostic and biomarker, next drug discovery and development, Next, personalized medicine. Next, agriculture and biotechnology, system biology, biomarker discovery, drug development, personal it is repeated, and next, cancer. In all of these, proteomics bioinformatics plays a most important role. Okay, please follow at LS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and also get it on Google Play Store. Thank you so much for watching my video. Please like, share, and subscribe at LS Academy. Thank you.